The use of DNA fingerprinting in forensics. This is for BCM 201 Assessment Task 1 by Charlotte Robinson, Nicole McGrath, and Cassidy Berger. So what is DNA fingerprinting? Deoxyribonucleic acid, which is otherwise known as DNA, contains the genetic information that makes up an organism. It is unique in every individual and therefore a very valuable tool in forensic science. DNA fingerprinting compares the DNA found in a person's nucleic cells with the DNA identified in biological matter found at the scene of a crime. This is done in order to identify or exclude that person. DNA fingerprinting can also be known as DNA profiling, genetic fingerprinting, and DNA typing. So the history of DNA fingerprinting. DNA fingerprinting was discovered by Alec Jeffreys at the University of Leicester in 1985. He quoted, "And on the morning 9:05 of Monday, the 10th of September, 1984, we got our first truly awful DNA fingerprint, purely by chance. The x-ray showed extremely blurry, but he could see there was patterns in the DNA and a level of specificity. A few months later, the technique was refined and Jeffreys was able to see clear, unique DNA fingerprints. After Jeffreys' discovery, DNA fingerprinting was then able to be used in forensics. It was first used in forensics in 1987. 2 teenage girls had been raped and murdered on different occasions in England, in close-by towns. Semen was obtained from each of the 2 crime scenes. At the time, Richard Buckland was a suspect. He was able to be excluded and clear his name with the use of DNA fingerprinting. Another man, Colin Pitchfork, was matched using DNA fingerprinting. He then later confessed to committing both of the crimes. The development of DNA fingerprinting. Jeffreys' original technology is now no longer used in forensics, and there have been many developments in his original procedure. Many of improvements have been made to overcome the original limitations of Jeffreys' technology, such as changing the labeling from radioactive to fluorescent. As you can see in the diagram here, it shows the labels, which can be known as markers, and show on the different strands of DNA. As the technique has improved, DNA fingerprinting started to be introduced around the world and is now used as routine in majority of forensic laboratories.
How does it work? As mentioned by Charlotte, DNA fingerprinting in relation to forensics on a simple level is when a DNA evidence sample is taken from a crime scene and treated with restriction enzymes, which cut and define certain sites. This is done to establish a pattern of fragments, and this pattern is compared with the DNA from the suspect's DNA treated with the same restriction enzyme. If the 2 DNA profiles are a match, then this shows proof that the original DNA evidence sample comes from the suspect. On a more molecular level, there is much more involved in preparing and comparing both the biological traces found at crime scenes and that of the DNA of the suspect. Genetic fingerprinting, also known as genotyping or DNA profiling, with relation to forensics, focuses on short tandem repeat sequences that are found in all of us. A short tandem repeat locus is a specific location on a chromosome where a short DNA sequence is repeated many times in tandem. These loci used in DNA fingerprinting are often very short and vary in length. The original accurate length of a short tandem repeat, even one that has been traumatised or partially degraded, can, with the aid of the polymerase chain reaction, be successfully determined. the DNA sequences bordering each short strand locus are identical, except for rare mutations, in all humans. However, the short tandem repeat lengths at these particular loci positions are different for all humans. Hence, this is our own genetic individual fingerprint. Polymerase chain reaction primers are targeted to the bordering DNA to amplify the DNA across the short tandem repeat. These primers also contain a dye colour that assists in individual evaluation at specific loci positions. The products from the polymerase chain reaction are then subjected to electrophosphorus on a very thin polyacrylamide gel in a capillary tube. The peaks that are then produced as a result of this process allow the true length of that person's short strand repeat to be visible. Hence, their DNA fingerprint is identifiable. It must be noted, however, that there are two lengths noted in each short tandem repeat, as each person inherits a chromosome from each parent. Thus, two differing length short tandem repeats will appear, reflecting a chromosomal pair. This is a figure showing two different hypothetical samples taken. The first one being the evidence sample, so the sample that would have been taken at the scene of a crime, and the second one being the suspect sample, DNA sample. As you can see from the first one, there are peaks going all the way along, and these are basically the short tandem repeats, or indicating the length of the short tandem repeats. Looking at the second one, where the suspect standard, you can see at the same loci position and the same numbers that the short tandem repeats of the suspect match almost in length to that of the evidence sample. So were this a true crime situation, you would say that the suspect was at the scene of the crime. This table here shows a more simplified version of the previous slide picture showing the peaks. This is actually the measurements that were put into a table showing different measurements for different suspects and also what the actual evidence sample was. So from the first column, there is the short tandem repeat locus code. So as mentioned earlier, each loci has a code or a number to identify where it is on the chromosome. So then we have the evidence sample. As mentioned earlier too, there is two figures because of the two figures from different chromosomes from different parents. So the evidence sample has the figures there of the short tandem repeats. Then we look at suspect A and suspect B, and they have their figures as well. Now, suspect A, the figures do not match up for the short tandem repeats, but suspect B completely matches up. So therefore, this shows again that suspect B was at the scene of the crime, as the evidence does match up. Current applications of the technique. Currently, this DNA fingerprinting technique is used all over the world for varying reasons. The first and main reason we are looking at here is, of course, for forensic identification in crime scenes. Evidence collected at crime scenes is analysed in a laboratory and often presented in court. When biological traces are found at crime scenes, they are an essential lead in identifying and convicting possible crime suspects. An application also using this DNA fingerprinting technique is familial DNA database screening or searching. This is based on near matches of close family relatives that have very similar, not exact of course, DNA sequences. An example of this case is in the UK in 2004, where a man by the name of Craig Harmon was convicted for manslaughter. His DNA was not part of the UK DNA database, however his brother's was. This similarity led the police to further investigate the family and finally convict Craig Harmon. This is also used for identification purposes. DNA identification using these methods have allowed victims of many disasters to be identified. For example, victims of the 9/11 terrorist attacks, people that have died in Hurricane Katrina, and also victims of recent wars. Perhaps not so much under the forensic banner, but still equally interesting, is the ability via DNA fingerprinting to track ethnic lineage. Many people are curious as to where they have come from, and they now have the abilities through these applications to find out. Advantages of the technique. There are many advantages of this technique, some that we have already mentioned earlier. The main advantage in forensics, however, is the extremely low chance of misidentification. When a sample of short-term repeat DNA is used, the chances of misidentification are less than 1 in 10 to the 18, or a quintillion, if that sounds more impressive. Also, an extremely small amount of DNA, only 1 nanogram, is required to obtain a genetic fingerprint. Therefore, only a minute amount of a suspect's DNA is needed to be left at the crime scene to be able to identify them. The Combined DNA Index System, or CODIS, database has been set up in the US to maintain extremely high level integrity of the DNA tests. All DNA tests performed in the US must use 13 strand tandem repeat loci to give the most accurate reading. Other countries such as the UK and Europe also have similar guidelines. CODIS has assisted in more than 274,000 forensic investigations to date. A main advantage, of course, is that this technique has given law enforcement agencies and courts the ability to both convict and acquit suspects based on DNA fingerprinting evidence. In the US alone, there have been at least 330 post-conviction exonerations based on DNA evidence, some of which had samples months or even years old.
Shortcomings of the technique. Several irregularities pertaining to the handling and analysis of samples and interpretation of data have surfaced recently during the course of trials involving DNA fingerprinting. They all fall into the category of poor scientific practice. Probably the most notable deviation frequently observed is the lack of appropriate positive and negative controls. In order to be sure that everything concerning the processing of samples and procedures is accurate, scientists include the comparison samples that are known to have the same positive control or different negative control fragment patterns as the samples being tested. If the suspect's DNA is processed in 2 separate batches, one as positive control, and separated in different wells in the gel, and the batches generate non-matching fragment patterns, then it is clear that something is wrong with the DNA purification procedure and/or the separation of the DNA fragments. Further shortcomings of the technique. An object with DNA can be placed at a crime scene in order to gain a conviction. This can be done any time before or during the forensic investigation of the crime scene. The use of DNA fingerprinting is very convincing and almost always leads to a conviction in a courtroom setting. Further shortcomings. The frequency at which mutations give rise to changes in the fragment sizes has been calculated. This type of mutation is thought to occur in the parent's reproductive cells, sperms and eggs. Another type of mutation that could be the basis of mismatch between tissues from the same individual results from uneven exchanges of DNA between partner chromosomes or the 2 halves of chromosomes. The phenomenon is referred to as somatic recombination, and it takes place in somatic tissue or non-reproductive cells. The net effect of somatic recombination, especially if it occurs early during the embryonic development of an individual, is to alter the DNA of 2 or more lineages of cells. These DNA alterations can affect DNA fragment sizes, resulting from restriction enzyme digestions. Different types of DNases including restriction enzymes from bacterial contamination of samples, are responsible for DNA degradation. There have been no cases of additional fragments generated as a result of degradation or cutting by the enzyme DNase 1. Yet, it is known that restriction enzymes and enzymes that digest DNA more indiscriminately, like DNases, do not cut DNA at random. In some cases, the site of cutting is very specific within certain sequences of genes. A potential cause of false DNA match is misinterpretation of the results. Laboratories sometimes mistype evidentiary samples. If the incorrect evidentiary profile happens to match the profile of the innocent person, then a false incrimination may result. Mistyping is unlikely to produce a false match in cases where the evidentiary profile is compared with a single suspect, but the chance of finding a matching person is magnified, or more accurately multiplied, when the evidentiary profile is searched against a database. A second potential cause of false DNA matches is mislabeling of samples. The best way to detect labeling errors is to obtain new samples from the original source and retest them. This safeguard is not always available. Evidence at crime scenes is typically cleaned up and thereby destroyed once samples are taken, and the original samples are sometimes exhausted during the initial round of testing. Retesting is rarely done, even when samples are available. Routine duplicate testing by forensic laboratories is another possible safeguard, but it too is rarely done. Potential future applications or extensions of the technique. DNA swabbing may become part of the arrest process. The arrestee would be swabbed back at the police station, and the DNA data can be added to the arrestee's profile in the database. This has the potential to lead to arrests and convictions of past crimes. It is possible in the future that DNA swabbing may lead to a positive or negative ID within 2 hours. This could lead to a decrease in investigation time. Evidence amplification through improvements of the process will lead to a more reliable result. Like anything, there is always a way to do it better. There is constant research into how to further improve DNA fingerprinting. Technical improvements could allow analysis of smaller amounts of DNA samples. Is the use of complementary RNA, cRNA, or antisense RNA probes as opposed to DNA probes. There are several advantages that cRNA probes have over DNA probes, which can lower the minimum amount of DNA required for analysis. Usually the use of cRNA over DNA probes increases the sensitivity of the assay by at least 1 order of magnitude. The poor laboratory practices mentioned before are good examples of difficulties that can be remedied just by enforcing good scientific standards. Others, like eliminating the uncertainties of band shifting, degradation, and inappropriate databanks, necessitate the generation of additional data or new technology. In terms of databases, it is clear that more basic research is needed to determine how genetically homogeneous specific human populations are and how heterogeneous the species is when broken into racial or ethnic clusters. For example, it is not known if Puerto Ricans and Mexicans are sufficiently different genetically to require the establishment of independent databanks. Also, it is not known whether specific ethnic groups in the United States, or in any other country for that matter, have intermingled with other populations to an extent that warrants unique databanks. These considerations can easily lead to requests for DNA typing of racial and ethnic characteristics.